Hello everyone, this is S.Bhargavi studying B.Pharmacy first year in MAHARAJAH'S College of Pharmacy. I am in front of you to discuss a few words on my topic glycogen storage diseases. First of all, before knowing about glycogen storage diseases, we should know what glycogen means. Glycogen is a multi-branched polysaccharide of glucose. That means many glucose units, that means more than 5 or 6. They combine or branch to form a glycogen molecule. We can see that in the structure of the glycogen. In the structure of the glycogen, you can see more than 6 molecules or 5 molecules of the glucoses which are branched or united to form a glycogen molecule. The importance of glycogen is it is the stored form of glucose which can be used when the body requires energy. Hence, we can say it is the energy storage form of glucose or energy storage form in the animals, fungi, bacteria, etc. We know that in plants, starch is the stored form of energy which is used by the plants. In the same way, in human beings or animals, fungi and bacteria, the glycogen is the stored form of energy which is similar to that of the starch in plants. And now we got to know what is meant by glycogen and the importance of glycogen. But where it is stored? The glycogen is stored mainly in two places, that is liver and muscle. In liver it is stored up to 6 to 8% and in muscle 1 to 2%. But due to more muscle mass in muscle, the quantity of glycogen is 3 times higher than that of in the liver. That means in muscle 250 grams and in liver 75 grams. Okay, now we said that the glycogen is stored in liver and muscle. But how it is stored? It is stored as granules in the cytosol of these tissues. Why it is present in cytosol means because the cytosol is the main site where we can get all the enzymes required for the glycogen synthesis as well as breakdown of the glycogen. That is the process of glycogenesis and glycogenolysis. Hence, it takes place in the cytosol of the liver tissues, muscle tissues, etc. Now we got to know the importance of glycogen and the storage of the glycogen. Now, what is the function of the glycogen? We said that in liver and muscle the glycogen is stored. Now, the functions are the liver glycogen is used to maintain the blood glucose levels. When there is a low blood glucose levels in the body, the glycogen breaks down into glucose until it attains the normal level. If there is low blood glucose levels or if there is high blood glucose levels, the glucose will be converted into glycogen and stored in liver and muscles to maintain the homeostasis. Hence, this is the function of the glycogen. But we said that it is the energy storage form of the glucose. Hence, we can also call it as a fuel reserve to our body. Why do we call as a fuel reserve means? Due to 3 reasons. First one is the glycogen is rapidly mobilized. That means it can be transported from one tissue to another tissue or one cell to another cell easily. Second reason is it can generate energy in the absence of oxygen. So it does not require oxygen to supply energy. And the third main reason is the brain requires the continuous supply of glucose for energy, which is mainly obtained by the glycogen. Hence, these are the 3 reasons due to which we call glycogen as a fuel reserve to our body. Now we got to know what is glycogen, the importance, the functions and the reason why we call it as a fuel reserve. Now we'll know about the glycogen storage diseases. If there is any metabolic defects that is concerned with the glycogen synthesis or breakdown, that is glycogenesis or glycogenolysis, then the disorders or the defects is called as the glycogen storage diseases. It is characterized by the deposition of the glycogen, that means accumulation of glycogen. Now we will get into the diseases. First one is von Gierke's disease. It is caused due to the deficiency of glucose 6-phosphatase enzyme which is required in the process of gluconeogenesis, that is during the transformation of glucose 6-phosphate to glucose. If there is a lack of this enzyme, then the glucose 6-phosphate does not convert into glucose and hence there will be the accumulation of glycogen in the form of glucose 6-phosphate. So there will be no energy supply to the body as there is no conversion of glucose. And it mostly affects the organs such as liver, kidney, intestine by affecting the renal cells and hepatic cells. As they affect the renal cells and hepatic cells, it causes the enlarged kidney and livers. The consequences of these diseases are fasting hypoglycemia, ketosis, lactic acidemia, hyperlipidemia. Fasting hypoglycemia means the low blood glucose levels in the body during the fasting conditions of our body. That means when we do not take any food or carbohydrate source. Ketosis means when body lacks glucose for energy, the fat that we contain in our body will be burned and made into ketone bodies which supply energy to our body. The ketosis can be identified by three symptoms: bad breath, and we can see weight loss, and appetite suppression. That means we will not feel any appetite or we don't want to eat anything. Lactic acidemia means the accumulation of lactic acid in our body. As there is no conversion of glucose, the glucose cannot get into the glycolysis cycle and there will be no conversion of lactic acid again. So, the lactic acid accumulates in our body. Hyperlipidemia means the more amount of lipids will be accumulated in our body. Coris disease, second one. It is caused due to the deficiency of amylo alpha 1,4 glucosidase. That this enzyme is required for the debranching of the glycogen. As there is no enzyme for debranching, the glycogen will not break down into the glucose and hence no energy supply will be there to the body. As there is no conversion or debranching of the glycogen accumulates in the body, mainly in the parts of liver, muscle, heart and leukocytes. As there is no debranching, the branched chain glycogen will be accumulated all over the tissues such as these affected organs. And it also leads to the enlarged liver, which affects the liver functions also. And the clinical manifestations are similar to the von Gierke's disease, that is fasting hypoglycemia, lactic acidemia, ketosis, etc. But the consequences or the manifestations are milder when compared to the von Gierke's disease. That means this is not much severe when compared to the von Gierke's disease. You can see in the picture in von Gierke's disease, the liver is enlarged and due to the glycogen accumulation, due to that kidney enlargement also takes place. And you can see in the liver hepatomegaly, that means hepatic cells will be increased, that is in hyperplasia condition, which leads to the distended abdomen also. That means prolonged or enlarged abdomen also. And the face will get doll-like face because of the accumulation of glycogen in the tissues also. And you can see the hypoglycemia, which leads to seizures also. That means electrical connectivity between the cells will be lost. And it also leads to coma, intellectual disability when it is severe. Now we'll get into another disease. Anderson's disease. It is caused due to the deficiency of glucosyl 4,6 transferase, which is a branching enzyme. That means glucosyl 4,6 transferase is responsible for the branching of glucose. So when there is no branching, the glycogen molecules, that means less branched glycogen molecules will be accumulated in the most of the tissues in our body. That means both epithelial, squamous and every tissue of the organ. It mainly leads to the liver cirrhosis, which causes impairment in the liver functions. That means liver do not function properly. And next, the Anderson's disease is also called as amylopectinosis, which is the other synonym for the Anderson disease. Now you can see in this picture the less branched, that means only this branch of the glycogen will be accumulated. That means the broken down parts of the glycogen will be accumulated all over the tissues. Next one is McAlder's disease. This is also called as type 5 glycogenosis. That means glycogenosis. Generally we have 5 types, in that the six types, in that the fifth type is the McAlder's disease. It is due to the deficiency of muscle glycogen phosphorylase, which is also called as myophosphorylase. Myo here indicates the myoglobin protein which is present in the muscle. When there is a deficiency of this myophosphorylase or muscle glycogen phosphorylase enzyme, there will be no removal of alpha 1 4 bonds, which is the glycosidic linkages present between the glucose molecules in the formation of glycose. As there is the no removal or degradation of these bonds, there will be no initiation of glycogen breakdown. That means the glycogen will not initiate the process of glycosenolysis, which generally results in the formation of glucose. But as there is no initiation of breakdown, there will not be formation of any glucose and glycogen accumulates. As there is no process of glycosenolysis, glycogen is not metabolized, which results in the ATP shortage. There will be no production of ATP as there is no synthesis of glucose. As there is ATP shortage, no formation of glucose and no lactate or pyruvate. If glucose forms, then only it enters in the glycolysis pathway, and then it converts into lactate, and then it is converted into pyruvate. Then during exercise condition it will turn into lactate. But as there is no formation of glucose, pyruvate and lactate are not formed, and during strenuous conditions or exercise conditions we cannot get supply of lactic acid, which is required for supply of energy, and it leads to the muscle failure or injury. And due to inadequate energy supply to the muscles, it also leads to the muscle cramps. That is, we cannot do our exercise properly, and if we do also, we will get some clinical consequences like muscle failure or muscle injury in our body. So every now we can conclude that every glycogen storage disease is generally linked to the or related to the glycogen accumulation, which is resulted due to there is no formation of glucose. Hence we can say the main reason is the glycogen accumulation. Almost all the organs are affected, but mainly liver is affected. We can see that among the 4 diseases we discussed, the 3 diseases are mainly linked to the liver. And enzyme specificity. That means every disease is concerned with a specific enzyme. That means we can see von Gierke's glucose 6 phosphatase, Coris disease amylo alpha 1,4 glycosidase, Anderson's glucosyl 4,6 transferase, McAlder's myophosphorylase. Like every disease has a specific enzyme deficit which leads to the certain concerned disease. And the disorders mainly depends on the glycogen metabolism, that is glycogenesis or glycosenolysis, either the formation or the breakdown of the glycogen. This is the main reasons or the main aspects which leads to the glycogen storage diseases. Hence, I think you got to know about the glycogen storage diseases clearly. I hope you all understood my topic, and I took this topic from as a reference book from U. Satyanarayana and U. Chakrapani. I hope all of you got clear about this topic. Thank you everyone.